Okay, so our lab group did the effect of temperature on enzyme activity, and the enzyme we used was trypsin, and trypsin is found in the small intestine of animals, and it helps facilitate the breakdown of proteins, and in this case it helped facilitate the enzyme reaction. So our question was, how do different temperatures affect the reaction rate of trypsin? And our hypothesis was as temperature increases and decreases beyond 37 degrees Celsius, the activation energy the activation enzyme activity of trypsin will decrease. So our conditions that we used, we had warmer temperatures where the azocasein and trypsin was incubated, and then cooler temperatures, and we used those to see the difference. So our experiment showed that the enzyme trypsin was most effective at 57 degrees Celsius with an absorbance of 1.65 nanometers, and that was significantly different from the other samples according to our t-test. And our hypothesis was not supported because we hypothesized that trypsin would be most effective at 37 degrees Celsius, but we found that at 57 degrees Celsius it was most effective. Okay, so our question was, how do different temperatures affect the enzyme reaction of trypsin? Our hypothesis is as temperatures increase and decrease beyond 37 degrees Celsius, the enzyme will be less productive. And then our rationale for this was, in the human body, trypsin is known to function best at 37 degrees Celsius with a pH of 8. Therefore, it was hypothesized that the most effective rate of reaction for trypsin would be at 37 degrees Celsius. Okay. We added 500 microliters of trypsin and 450 microliters of reaction buffer to a microcentrifuge tube, and then we placed the tubes in corresponding temperature baths. So some are colder, some are hotter, to bring the tubes up to temperature. And then we added 500 microliters of azocasein to each microcentrifuge tube containing the trypsin reaction buffer. We then incubated each sample for 20 minutes in their corresponding baths, and after they were finished incubating, we added TCA to stop the reaction. We then centrifuged the tubes at 2 minutes at 14,000 RPM. We collected the supernatant and transferred it to a new microcentrifuge tube, and we centrifuged again for 2 minutes at 14,000 RPM, and then collected the remaining supernatant and transferred it to a spectrophotometer tube where we measured the absorbance and then we repeated the entire process for 2 and 3 trials. We centrifuged the tubes to remove the excess undigested substrates and trypsin, and then so the supernatant is the buffer and the sulfamylinide, and yeah. Okay, so our substrate was azocasein, and the enzyme trypsin cleaves azocasein, and then that leaves sulfamylinides in the supernatant, and the more orange the supernatant is, the more sulfamylinides are in the substrate, and then the more effective the enzyme trypsin was. Okay, so our control variable conditions included a constant pH of 8, the constant salinity, the constant time and speed of the centrifuge, and incubation time of 20 minutes. We had the controls so that we were making sure that we were only testing the effect of temperature on enzyme activity and not another variable. So we used negative controls. We had 15 because we had 15 trials. They were prepared the exact same way as the positive controls, but the negative controls didn't have trypsin, we added distilled water instead of trypsin, and that shows like any other things that could be affecting our results. So we had 3 replicate trials because that's how you test for variability and you reduce the number of outliers. Is it not? Look at how iconic that one is. Okay, so for our results, we had 2 outliers, one for 15 degrees Celsius and one for 70 degrees Celsius. But our hypothesis was not supported because we said at 37 degrees Celsius, it would be the highest activation energy or like enzyme activity, and at 57 degrees Celsius, we have the highest absorbance. So then these are our results continued. So we have standard deviation bars, and then just enzyme activity. These 2 showed that we had outliers, but the rest of them were good. And then for our t-tests, at 15 degrees Celsius, for positive and negative control, for negative control, it was significantly different, but for the positive control, there was no significant difference. For 25 degrees Celsius, positive and negative control, they were both significantly different. For 37 degrees Celsius, positive and negative control, they were significantly different. For 57 degrees Celsius, positive control, there was a significant difference, and then negative control, there was no significant difference. And then for 70 degrees, positive control was significantly different, and negative control was no significant difference. So in our discussion, the experimental design was adequate to test our hypothesis. The measured data had pretty consistent results except for the 2 outliers. We did have some difficulty with the pipetting. We had multiple people pipette, and we messed up the timing. So those are things that we would change in the future. And then our hypothesis was not supported because the results indicated that the experimental group of 57 degrees had a higher enzyme activity on average, and the hypothesis, we inferred that the control group of 37 degrees would have the most enzyme activity.